Hi, good morning, everyone. Not sorry, you're not here with us live. We have a great fun group together at the AVRC, but thanks for joining us online. I'm going to introduce Sara Gianella Weibel, a very dear friend of mine and an amazing leader, researcher, advocate. She's a leader in clinical and translational virology, dedicated to transforming the landscape of infectious disease research. She's the director of the San Diego CFAR Translational Virology Core and the ACTG Virology Specialty Lab. She co-directs the Neurovirology and Biology Core for the HIV Neurobehavioral Research Center and the HOPE T32 training program, reflecting her commitment to education and mentorship in the biomedical sciences. She's driven by a profound commitment to social justice, and she's devoted to enhancing the diversity of the biomedical workforce, enriching educational experiences, fostering scientific discovery and innovation, and addressing health disparities. She passionately conducts research involving cisgender women, transgender individuals, and underrepresented populations, striving to build public trust and maximize the positive impact of research on diverse communities. I can go on and on. I think a lot of people have heard Sara talk recently about the Last Gift program. She's going to pivot today and talk about some of the work that she's been doing through the ACTG, which I don't think we've heard very much about in a while. So very excited to have Sara take it away.
Yes. Thank you so much, Jill, and thank you, everyone. And yes, I was going to say too, I know that over the last year, everyone always knew me for Last Gift, but really, you know, what I have been working on for most of my life as a researcher is the work around CMV. This is how I started here 15 years ago, and kind of the topic that has followed me. And what makes me particularly proud, that I don't think it happens so many times in the life of a researcher, is when you start something at the bench and then you're able to bring it up to the clinical trial. And this is why I'm particularly excited and proud about the clinical trial that I am chairing. One is 5383, which is the one that I presented today. And the other one is 5355, which is the anti-CMV vaccine that is still ongoing. And hopefully we'll have some data probably by next year. Okay, let's give a little bit of a background. Like, we know that there has been a gap in life expectancy in people living with HIV that, you know, due to all of the progress in antiretroviral therapy has been progressively narrowed, but it didn't close completely. And we know that there is about still an average a 9-year gap in mortality. And when we look at morbidity, there is even a larger gap, right? And this is also true for people that have maintained immune system with CD4 T cells over 500. That means that we did a fantastic job with antiretrovirals, but still we have work to do, right? To make sure that people with HIV have the same quality of life and like a sweat vaccine. And this is why there is a big push right now to find ways to close this gap even further. So we do know that inflammation is one of the main driver of morbidity and mortality in people with HIV, even when they are treated on antiretroviral therapy. I think everyone is familiar with the SMART study, which now it's been done a while ago, that kind of really hinted that inflammation was a big driver of morbidity and mortality. And since then, this has been confirmed by many other studies. Really, the field's goal right now is to try to move the people from the top quartile, the red and the orange, down to the lower quartile, right, by decreasing inflammation. So inflammation is bad, not only for people with HIV, but also with people without HIV, right? These are some examples of studies that have been done where interleukin-1 beta, for example, has been shown to be associated with cardiovascular disease and death. And there has been a study done a while ago where blocking interleukin-1 beta decreased cardiovascular events in people without HIV. And what's interesting is that it didn't only increase cardiovascular, but also cancer mortality. So I know that, cardiovascular is the main outcome that people think associated with inflammation, but really even cancer or diabetes, like all of these aging-related disease have an inflammatory component. So a lot has been done in people with HIV trying to reduce inflammation. And for those of you that are familiar with the ACTG, there is a whole group of people who's called comorbidity TSG, that the whole mission is to try to reduce inflammation. And I'm part of it as a virologist, and every month there is a new idea about a new compound that we can try. These are just some of the ones that have been tried, but really so far, nothing has really worked, except, and then I will talk about it. And then there is a few that show promise recently. One is Ozempic, and the other one is aldosterone antagonist, where they were recently presented. I think everyone knows about Ozempic, that right now it's really everywhere, and it looks like it's helping with a lot of morbidity on the top of losing weight. And then Reprieve, right? Like Reprieve was kind of a breakthrough, where this was a super large study that has been done as part of the ICTG across the whole world with thousands of people enrolled, and then it was terminated early because they were able to demonstrate that putting people on statin and pitavastatin in particular, even if people didn't qualify really, like even people that technically had low risk for cardiovascular disease, was decreasing the cardiovascular risk by 35%. So such a big decrease that they ended up terminating the study early and then putting everyone on the statin. So it's important to note that in Reprieve, the effect was on cardiovascular disease. There was no effect on other aging-related disease. For example, there was no effect on cancer. And if anything, there was a slight increase in diabetes, which is known, by the way. And another thing that is important to know from a mechanistic point of view, the statin effect was linked to decrease in LDL and not so much inflammation. And I think this is important because it shows that there is still room, for improvement, right? Like this is one mechanism, but it didn't address the inflammation. In fact, there was no reduction in CRP and interleukin 6 in the statin group versus placebo. So this is why, we are not done, right? We can still continue to do more research. Like 35% decrease in cardiovascular disease is amazing. And that's probably the most successful study that ACTG has done in a while, in the aging field for sure. But we can probably try to make it even better with treating something else, and in particular, some of the driver of inflammation. And cytomegalovirus has been suspected to be one of the main drivers of inflammation since a long time. And to give you some background, this is a study that I have done with Sheldon Morris a very long time ago, where we basically took seminal plasma from PERC participants. These are people that were, treated early during HIV and then followed longitudinally for a long time, and they were donating semen regularly, and we measured CMV and all of the other herpes viruses. And we, despite the fact that these people had conserved immune system and they had relatively high CD4 T cell, as you can see, they have almost everyone had at least some CMV and EBV shedding in semen. This means that really there is a big subclinical shedding, because these are all people that were completely asymptomatic, but still they were shedding CMV and EBV. And we know that CMV has a big impact on the immune system. Like CMV has been evolving with us since millions of years, and really has become very smart on how to, trick the immune system. And this is why it keeps replicating in the body, and the immune system keeps trying to, keep it under control. And over time, as people age, a big proportion of CD4 and CD8 T cells are engaged in battling CMV, right? And these are data from people without HIV, and you can see that we're talking about up to 20%, 40% of memory CD4 T cells, in average it's 10%, are fighting against CMV, are anti-CMV specific. Like, this is huge if you think how many pathogens a person sees over their life. No other pathogen is able to engage the immune system in this way. And when we look at general inflammation, like not CMV specific, we can also see, and these are data from a paper that we published with Mike Freeman at Case Western 10 years ago. Like, these are people people without HIV, and then people with HIV with and without CMV. And you can see that people with CMV and HIV had higher level of CD8 T cells and higher level of soluble TNF receptor 2. And keep that biomarker in mind, because this data is what we use then to design our clinical trial. But you can see that it looks like CMV has almost a bigger effect on the immune system than HIV. So there have been a lot of observational study linking CMV to a surrogate marker of CMV to treated HIV. And when I say surrogate marker, it's because some of the study look at CMV DNA, but many study look at CMV IgG because it's an easier to measure, and people assume it's a surrogate marker of CMV activity, which now we know it's probably not really true. But you can see, right, CMV DNA has been associated to AIDS-defining events, CMV IgG to non-AIDS event, anti-CMV response to arteriosclerosis, cardiovascular disease, SCOT, CMV IgG, neurological complication, cancer, and larger HIV reservoir. Here I wanted to show you some of the main study. Like, this is the ICONA study, where they showed association between being positive for CMV IgG and non-AIDS related death with a very strong effect on cardiovascular disease. This study was done in Italy 9 years ago. This is an early study done by Priscilla Shu at UCSF, showing a correlation between CMV-specific CD8 T cell and carotid intima media thickness as a marker of cardiovascular disease. So again, association between CMV-specific T cell response and marker of cardiovascular disease. This is another study, more recent, showing a correlation between CMV IgG titers and cancer risk in people with HIV who are treated. And then these are mice, but there is some evidence that maybe CMV might also be associated with metabolic disorder, inflammation in the fat, and diabetes. And there's a whole other line of investigation that we are following up now with some colleagues of us at Vanderbilt who are kind of really interested in diabetes and fat. And we're working together to see if what we see in mice might also be true for humans. So this is the Scott's paper where we see that people with high CMV IgG had worse neurocognitive impairment compared to people with lower CMV IgG. By the way, this is one paper. This has been confirmed by other studies as well. And this is an old study of mine showing a difference in people with and without CMV shedding in HIV reservoir and proliferation of CD4 T cells. So this is basically to say there is a lot of cumulative evidence that CMV might be associated with inflammation, with some morbidity, but until now, it really was mostly observational. But really, the chicken and the egg question always stays, because people can just argue that if you have a lot of inflammation, you are more likely to shed CMV, just because a lot of viruses like to reproduce in a more inflamed immune milieu. So the only way to really definitively understand if CMV was a driver or just a bystander was to do a clinical trial. And in 2011, Peter Hand did a first small clinical trial. It was randomized. At that time, he used valganciclovir. It was a relatively small sample size. So I think it was 30 patients, 30 participants. And also not everyone was completely suppressed. And he did observe like a decrease in CD8 T cell activation in people that were on valganciclovir. And what is interesting is that the effect remained when people went off drug for at least 4 more weeks. So while this was a small study, it was really like a proof of concept study. And the reason why we didn't go bigger with valganciclovir is because, as many of you know, it's not a very safe drug to take for people that don't really need it. And it has side effects. And so we didn't think it was scalable to give valganciclovir to a lot of people. But this was definitely like a proof of concept that maybe treating CMV was a good idea. So we went back and because some plasma was available from the same study, and Peter in his lab ran some of the biomarker, one of them being soluble TNF receptor 2, but he also ran CD163 and CD114, and both 163 and 14 are marker of monocytes activation. And you can see that there was, a significant decrease in all of them, right? Like really impressive. And so based on what we know of these biomarkers, if these hold true, a decrease in soluble TNF receptor 2 of this magnitude will correspond to a 22 decrease in myocardial infarct and stroke, and up to 50% decrease in diabetes mellitus. So impressive, right? And if you think that this might be cumulative with statin, we're really excited about it. But another reason why valganciclovir was not the best drug to test this hypothesis is that valganciclovir is a pan-herpes virus drug. So it doesn't suppress only CMV, but really suppress every other herpes virus, right? So there's really no way to isolate the CMV effect. And maybe it's EBV, maybe it's herpes simplex, like who knows? So this is why when finally Merck came out with Letermovir, Peter and I ended up designing this study, right? This is 5383. And the primary hypothesis was to assess whether Letermovir therapy for 48 weeks reduces plasma soluble TNF receptor 2 level, which we selected as the primary outcome, and other inflammation and cardiometabolic indices versus no anti-CMV treatment in people with HIV. To tell you a little bit of the story of the Letermovir trial, it started a long time ago. And as often happened with the ACTG, it got stalled for years because everyone had something to say, and then we had to keep going back and renegotiating. The initial trial was a randomized placebo-controlled trial, but the ACTG stalled it so long that Merck basically stopped producing the placebo because they were done with their own trial, and producing placebo is expensive. So they shut down the placebo facility and they say, sorry, and so we had to basically kind of take a compromise and redesign the trial. And now it was an open label, Letermovir versus no treatment, which, as you know, it's not ideal, right? Because now both the doctor and the patient and the study participants are not blinded. And so there is a lot of bias and confounding factor at play. And this is just one lesson learned about, we were really mad. And Peter sent a very strong email to everyone, really to, because by the time we started and the time we implemented, despite having probably 15 rounds of reviews, there was no big change made to the study other than losing the placebo. Like it was all small thing, right? But everyone feels so strongly about it that at the end, we wrote, like Peter wrote this email and he was on fire. And I was like, and I hope someone learned something about it, maybe. Okay, so how did we select the primary immunologic outcome? We put a lot of thoughts into soluble TNF receptor 2, right? It was not just the first that came to our mind, right? So we have data that soluble TNF receptor 2 increase in people that are seropositive for CMV, like that was our early paper with Mike Freeman. We know that it predicts cardiovascular disease, diabetes, and cancer. These are all data from the literature. It got suppressed by valganciclovir, like I showed, with the sample of the trial. And it's easy to measure. It's just an ELISA. We still had to go through the whole painful process of validating the ELISA, because every time you make an assay primary outcome in a clinical trial, the FDA requires that it's at least fit for purpose validated, which at the time nobody realized how painful it is. So we had a collaborator that has a lab, but he went through the whole process of validating TNF receptor and CD163 for the purpose of this trial. So just like a side note, if you ever design a clinical trial, be careful about what you put at your primary outcome, because it has consequences. This is the study design. So we had, the initial study design was 180 people. Everyone was pressed, seropositive for CMV, and 40 and higher, because we know that it takes a little bit for CMV to start having that effect. So we wanted to maximize the chance to see an effect. And half were randomized to Letermovir, 480 milligram daily, and antiretroviral therapy, of course, for 48 weeks. And then the other half was randomized to no anti-CMV treatment. And just we, by design, wanted at least half of the people with low CD4 and 1/3 of the people women. And I know that this put a big burden on the site, because these are all group of people that are hard to find, but we felt it was very important because there is, evidence that people with lower CD4 and women might respond differently to the treatment, right? So we really wanted to make sure we were able to do some stratified analysis with this population. And then we were following people for another 12 weeks while they were off Letermovir, but still on ART. And then we, this is kind of the schedule of evaluation where we were collecting blood, we collected saliva, semen, urine to measure CMV shedding. Then we were measuring immunological, virological outcome. And then we got like 3 one was the cardiovascular arrow one, where we wanted to add PET CTs to see if there was a decrease in aorta inflammation. There was another study that was adding fat biopsy, because we wanted to prove if really we saw a decrease in fat inflammation. And then we were supposed to do a neurosub study that NIMH was going to fund to see if there was a decrease in inflammation in the lumbar puncture. And then we had another NIDDK funded R01 to do gut biopsies because we wanted to see if there was a difference in inflammation and reservoir at the gut level. So a lot of work went into it and a lot of funding. And but the ACTG decided that it was a good idea to request a futility analysis after the first 40 participants enrolled. and after they reached week 8. And we can talk a little bit about the futility analysis idea. In retrospect, I think it was a big mistake. And I think futility analysis are a very good idea when the intervention is risky, because obviously you don't want to, more people to have a risky intervention if there is no benefit. But in this case, nothing really was risky, right? So the only reason why the ACTG did the futility analysis, because they were worried that this was a complicated study, because we wanted to enroll women and low CD4 T cell, and that we would just take a lot of resources from the site. But really, we had 4R1, right? So what they did is they transferred the risk from the ACTG to the investigator, because now if what happened, and you will see, if the futility was hit, all of our R1 basically, we were not able to get them. So again, I'm not very happy with this decision, but it is what it is. And at the time, we didn't know better. So we ended up agreeing with the futility analysis, and we had to pick a marker, right? We picked soluble TNF receptor 2 and soluble 163 as our futility marker. And so in the protocol, we write in that if after 8 weeks, 40 participants, we didn't see an effect, the study will shut down. And we were really sure we will see an effect based on all of our preliminary data. So these are the 40 participants. We enrolled 42, but only 39 ended up contributing to the futility analysis because one person had a prohibited medication, and so we had to exclude him. And we did have 45% with low CD4, 30% female, and this is the clinical characteristics. So there was no major safety signal. There was really no significant difference in safety between arms. That's good. So Letermovir is safe. But then when the results of the futility analysis came back, there was an unexpected increase in soluble TNF receptor 2 after 8 weeks. And soluble 163 didn't move, right? So the safety committee came together with the team, and the decision was made to basically shut down the study. So we were able to follow people off Letermovir for an extra month, but we had to close enrollment. And so this meant for us that, we had to go and renegotiate all of our R1. Thanks God I was able to keep mine, but Peter Hans PET-CT R1, he had to give back the money, which is heartbreaking. I know, that was really hard. And the FET one, we were able to repurpose. So, it was still kind of a very painful process. So after the initial shock, we started thinking, what can we learn? In reality, if this was from a pure intellectual perspective, this was actually a very interesting result, right? How could suppressing CMV increase the biomarker, right? So looking back at the literature, there is some evidence that CMV is not a straightforward guide. In fact, I don't know if maybe everyone remembers there was a big paper in Science a few years ago where they showed that having EBV was associated with increased MS. But then there is also a paper that showed that CMV is associated with the decrease of MS. And last year, Peter Hans' group at UCSF showed that having CMV was actually associated with a 50% decrease in risk of neurologic long COVID, while EBV was associated with an increased risk. So it looks like CMV is not as straightforward. And there is a difference. There seems to be a different immune effect between CMV and EBV. And so basically after doing research, and so what we ended up is interleukin 10. So interleukin 10 is an immune regulatory cytokine that everyone is producing, right? And the job of interleukin 10 is induced by inflammation, and then it reduces inflammation. It's the way of the body to it acts like a brake of the inflammation in the body. So like because the body needs inflammation to fight, some diseases, but then you don't want the body to go in a cytokine storm, and otherwise you die, right? So interleukin-10's job is to try to regulate the inflammatory milieu. So it comes up, and then it kind of really acts as a negative regulator of inflammation. And what's interesting is that both EBV and CMV make their own IL-10. And there is a CMV IL-10 and the EBV IL-10, and they look very similar to the human IL-10. It's their way of the virus to hijack our immune system, right, and create a milieu that is favorable for them. But what is interesting is that the EBV IL-10 is a partial agonist, and it's weaker than the human IL-10. But the CMV IL-10, it's a super agonist, and it's more potent than the human IL-10. So basically, when you have CMV IL-10 on board, it's stronger than your human IL-10, right? So maybe the CMV IL-10 was kind of keeping the inflammation down. There's also other evidence in people that have polymorphin at the gene level with the IL-10 receptor, and people that are, that have a polyformase that is associated with increased IL-10 receptor signaling are associated with more risk of CMV retinitis. On the other side, polyformase associated with decrease in IL-10 expression is associated for increased risk of MS. So it looks like IL-10 might play a role here. And so we went back as we measured IL-10 in the Letermovir plasma. And as you can see, human IL-10 didn't move, but the IL-10 receptor, which is part of the IL-10 pathway, significantly decreased. We were not able to measure the CMV IL-10 in plasma because that's not where we expect it to be. I think CMV replicates more in tissues. And so we were unable to measure the CMV IL-10, but this, like no change in human IL-10 with decrease in IL-10 receptor is indirect evidence that maybe IL-10 is really part of the mechanism, right? And so maybe when you decrease CMV, you kind of screw up the balance, and maybe there is an effect at the beginning where IL-10 goes down and the inflammation goes up. But now, since they shut down the study, we will never know. Maybe if we go longer at some point, the inflammation comes down again. So what we did then as a next step to try to really understand the whole picture a little bit better, we ran proteomics on the Letermovir samples. And the proteomic, like Olink has this proteomics panel that are cardiovascular and inflammatory panels. And you can see that basically a whole bunch of marker decreased in the Letermovir arm. And a lot of them are associated with inflammation and endothelial dysfunction. So there was a huge Letermovir effect. Somehow we were just unlucky, and the biomarker that we choose for our futility analysis was the one that went the opposite way. So I think that's another lesson learned, right? That when you design a futility analysis and you base it on one biomarker, it might not be a good idea, because really the immune system is so complicated that there needs to be more than one.
But you guys were sort of forced to do that.
Yeah, we were.
She's being very gracious.
I am not. So what was even more exciting is that there is this paper from last year where it was a case control study, and they did the same Olink proteomic marker, and then they used Mendelian randomization, which is a statistical method to try to infer causality between some marker and cardiovascular disease. And this study found 5 hits, and 3 of them were significantly decreased in our Letermovir trial, right? So this was even more exciting because So based also on the literature, it really looks like the proteomic landscape and how it looks like might be favorable. We also were excited to see that Letermovir convincingly decreased IL-1 beta. If you remember at the beginning, I showed you that this was a marker that was significantly associated with cardiovascular disease in people without HIV. Letermovir suppressed CMV mucosal shedding. So we know now that CMV had an effect, also like that Letermovir suppressed CMV. So we didn't have any breakthrough. And we measured it, and yes, genital, rectal, oral. No effect on EBV, because that was another thing. I was thinking maybe there is like a strange effect on Letermovir on EBV, and maybe, you know, something happened with EBV, and this is why we didn't see the decrease. But then if you see EBV, really there was no effect. So EBV doesn't seem to explain this strange result. So the conclusion is that treating asymptomatic CMV in people with HIV on antiretroviral therapy is safe and well tolerated. There was this unexpected increase in soluble TNF receptor 2, but we also saw a broad reshape of the plasma cardiometabolic proteome, which is different to the statin. So this will be like a synergistic effect, right? If we can treat CMV and give a statin, there will be like a cumulative decrease. It did suppress several plasma biomarkers that we know were causally associated with CMV. It suppressed, for example, IL-1 beta, which has been associated with cancer and cardiovascular disease. And so I do think that we're not done with CMV, so really we should go back and probably design another study. We're waiting now to, finalize the analysis, write the paper, and then we will have to go back and renegotiate with the ACTG or whoever wants to fund the study. So additional consideration is that similar to the other trial from Peter Hand, the suppression of CMV was sustained even when Letermovir was stopped for at least 12 weeks. So mechanism is unclear, but it's probably worth investigating. I think it's important because if we really see that treating CMV is a way to improve the quality of life in people with HIV, maybe we don't have to put people on Letermovir forever. Maybe there is you can do like a pulse effect, like treat them for a while and then go off and on and off. So the differential result between Letermovir and valganciclovir study might be because valganciclovir was suppressing more herpes virus, but I know that Peter Hunt is submitting right now in R1 to try to understand a little bit the difference between the two. And there is a risk of whack-a-mole effect. We kind of knew that, but that when you target one thing, the other one comes up, and maybe we really need to give the immune system a little bit more time to balance before we draw conclusion, right? And I do think that targeting CMV seems to be a safe way to reduce interleukin-1 beta and IL-6 signaling on the long run, but obviously this needs to be tested now in a new trial. And upon reflection, it appears that conducting a futility analysis in this time was really not a good idea. Okay, and then so a lot of people, were part of this story over many, many years here at UCSD, but obviously this is me and Peter Hunt with our CROI poster that was accepted as a late breaker last year. And a lot more work is planned. And I think we learned a lot. Like, I know I'm mad because of how it went down, but I do think that we learned a lot. And I do think that someone will have to fund a follow-up study based on this. Yes.
Thanks, Sarah. And thank you for telling us about your experience. I think really the entire audience can learn from it. So I guess, how do you envision, because it's interesting the way you brought up the statin story. To this day, we have a lot of hesitation from people using a daily statin. We can tell them all about the trials about cardiovascular prevention, but yet it's a daily pill. that causes muscle pains and all of that. So with Letermovir, so we can, if they're asymptomatic, we can tell them all about the prevention effects of cardiovascular risk of just treating the CMV, but it will be a daily pill, right? How do you envision, do you think Letermovir, are there other agents coming along the pipeline that maybe could not be a daily pill or?
So I think if we can convincingly prove that there is a market, people might come up with not daily pill, or maybe they can combine it with antiretroviral therapy or do, I think that right now nobody has done it because, but if you can imagine, if you can convincingly show that, everyone that is living with HIV can benefit from a particular therapy, I can see the pharma industry to run with it. And, it will take some time. I don't have the perfect idea, obviously, right?
Put it in your once every 2 month shot with your statin and your antidepressant and your
But I think Gabe's point is really important. I mean, I think we have really compelling data from Reprieve, and I don't think they're easy conversations to have with patients who are like, I'm trying to reduce my pill burden. Now you're telling me I gotta add something else. And I think I think separately, just the statin industry, right? They still haven't come up with an injectable, a long-acting injectable for a medication that really so many people can be on outside of, the HIV landscape. There hasn't been a move in pharma to do that. So I don't mean to be a downer or anything, but I do recognize this being complex.
Yeah, yes, for sure. And I don't know, Scott, being myself not a practicing physician, I feel, I don't have the perfect solution, but do you have anything?
So I was still I'm thinking about my grant application and clinic, so I didn't hear what Dr. Blumenthal asked.
I didn't ask anything.
Yeah.
I was really agreeing with Gabe, and even just the motivation of pharma to do things like, let's deliver statins in a different way, and they haven't done that,
Sarah and Peter have done a really nice job about framing the environment, and Peter often shows the tree picture and saying, there's a lot of branches and a lot of roots. What we have to find, it's not politically correct to talk about cutting down trees, but if you want to cut down the tree of inflammation, you don't want to clip the branches or just chop a root here and there, or have to cut multiple roots. You want to cut it at the trunk. And so, maybe one way of approaching that is to say, we'd have to prove this, but if you treat CMV, you won't need a statin. You won't need an antihypertensive. You won't need.
Well, then pharma will be really angry.
Well,
if we take statin.
But they could just charge more for that, as well, all the others, right?
Yeah.
And so, I mean, it has a way
Right.
of, the cost has a way of working itself out, but, in terms of selling it to patients, right? If it was proven that you can take this one pill once a day, or maybe in 5 years, we have a once a month shot, you can spare yourself all these other issues, insulin resistance and weight gain, right? I mean, all these other cardiometabolic abnormalities, if they are truly linked to CMV, as Sarah's nicely shown that they appear to be, and we treat CMV, do we spare people all those other conditions and therapies? So in the end, you take fewer pills,
Sure,
but that's. There
you go.
Yeah.
thank you.
And it may not be everybody too. I think we see the people all the time that have those persistently crazy high CD8s. And I know my patients are always like, why is this so high? What's going on? And we don't have a lot. And we do have a lot about the CD4, CD8 ratio being a marker for mortality and increased morbidity. And we don't know for sure, but hopefully we'll find out. But, maybe targeting CMV does reduce CD8 activation in some people. So maybe the people who have those really skewed ratios could be the people that need that, maybe not everybody with CMV. And vaccinations are
But it's a larger issue, right, outside of people with HIV. I mean, that's always
Sure.
been one of the promises of HIV research is what is generalizable to the general population. And people who have receptive sex have more herpes virus infections, including CMV. And they increase the more shots on goal you have. And so the longer you live, the more herpes viruses you get. You know, and so women, particularly women of color and Latinas have more CMV as they age, and it diverges, the curves diverge. And so, you know, it really is sort of an issue. I don't want to give my own lecture here. It's an issue outside of HIV. So, I mean, the case can be made about, you know, protecting vulnerable populations,
Sure.
and that is just not, it's not just a problem for people with HIV, it's a problem for everyone.
Yeah, and there's a big socioeconomic factor to CMV, right? So it is definitely an intervention that will benefit otherwise underserved people. So
And I was just I was hoping maybe others would say, I have no experience with a Letermovir. It seems, I mean, we're using it for patients with solid organ transplant and bone marrow transplants, and I just quickly said, how's that going? And it sounds like it's incredibly well tolerated, which is great.
we didn't have any breakthrough, we didn't
What'd you
have
say?
any resistance, yeah.
Which was great, because resistance, is, I think, the main,
Yeah,
but you guys didn't see that.
No, we didn't see any breakthrough and any resistance, no.
but again, I think also going back to just, will you take this medication? It has to be something where people aren't saying, wait, what about these muscle aches that I started to get, which I really do think has been, yeah, that's what I was talking about, just tolerability, but obviously there are other
Yeah.
considerations to have, like resistance. Yeah.
It is complicated, right? It's not a easy story, and I do really believe that the Letermovir trial has added a huge knowledge to the CMV field, and I think the IL-10 story is a robust story. I'm excited about it. IL-10 has been investigated also for cure strategy. I know that there's a few papers coming out. One is not published yet, but on biorepository from Rafik Sikali with the non-human primate model where they showed that combining IL-10 and anti-PD-1 decreased significantly the reservoir. And there was a CROI poster from Mirko Pagliardini on the same line, where monkey that received anti-IL-10 had a delayed viral rebound. So IL-10 right now is really hot. So this is why I feel like this kind of really comes at the right moment. And the other one that I just wrote this summer, there was an RFA about comorbidity, co-infection, and cure was all around IL-10, CMV, and so I think there is a compelling story here. So I'm excited about writing this paper. We're now finalizing all the data and writing a paper, and then when the paper is out, hopefully we can. I know Peter has been having conversation with Merck, and they seem amenable to, kind of donate Letermovir again. And we will have to reapproach the ACTG and see if they are willing. I feel maybe they feel guilty. And now, because my feeling is that the study will look a lot like the one, because I think that now we can really, with this evidence in hand, the next study, we can really start looking at endpoints like arterial inflammation, neurocognitive impairment. I'm excited by this. And now I'm excited even to see what happened with the 53-55, the vaccine, because obviously if we can see if the vaccine works, then that's a different story, right? That's even more exciting than the Letermovir. We'll see.
Are you, I should know, but I forget, are you looking at the vaccine people who don't have CMV or people who already have CMV?
No, they all have.
Therapeutic.
Yeah.
Therapeutic.
Yeah. Any other questions or?
Hey,
Jeff,
Sarah, sorry, can you hear me? This is Sanjay.
Hi.
Yeah. Hey, did you guys ever think about looking at the CMV quantiferon responses in your patients? I forget what it's called, the actual test, but it's essentially like a quantiferon test for CMV.
Well, I don't think we have, like, we
So-
definitely have planned to look at CMV-specific immune response. Yes, that's part of the R01, Sanjay. Now, let me,
Okay,
the R1 that we just submitted right now, we're planning with, I have a collaborator at UC Davis, which is an immunologist, and we are planning to look at, she's also the one who was leading the gastrointestinal R01 for 5383 and 5355 to measure the CMV-specific immune response in blood and in the gut. So yes, definitely.
yeah, it'd be interesting to look, pre and post therapy and how that affects some T cell responses, and then, anyways, cool.
Yeah.
Yep.
And IgG too, right? Like that's an easier one, but yeah.
Rachel's done a lot of work like that though, too, with CMV,
Yeah.
back in the day.
Yeah.
So.
What did this Jeff Taylor say?
He's saying a lot. I don't know if Jeff, you wanna unmute yourself and share?
Sure. Well, I was just pushing you back against the notion that, people aren't gonna take a pill. And I think especially for older patients who are the ones at greatest risk, who have the most to gain from this, I think if it's a small, easily tolerated pill, it's not gonna be such a big deal. And I sit in a lot of conversations with pharma where they say, oh, well, we're past pills, we're all about injectables. And a lot of people don't like injectables. They're fine with taking a pill if it's a small, easy to take pill. And I think, there's a lot of movement being made, at least in antiretrovirals, where it's like once a week and once a month oral regimen. So I think there's some possibilities there. But yeah, I just hate to hear people say, oh, it's just not going to happen because patients aren't a monolith. A lot are fine with that. And sure, there's people who don't want to take one more pill for anything, but I don't know what you do with them.
And can I follow on that? I'm not sure it's always about the pill. People don't want to have a disease. People don't want to have a lot of things that the pill represents. So I think, you have folks who aren't wanting to take pills for a variety of reasons. And maybe only one of them is I just don't like swallowing a pill every day. There's a whole other body of meaning out there. And of course, some of it could be related to depression, but there are a lot of other kind of experiences people have throughout their lives and experiences that they have currently that influence their health-seeking behavior.
And then a
And
question,
go ahead.
just a follow up for the practicing clinicians. What has been the uptake of pitavastatin? I mean, that's a good example of a small, easy to
can tell
tolerate
you.
pill.
None, because we can't prescribe pitavastatin because it's not covered by insurance. Sorry, I'm gonna say that, and I know we're not prescribing pitavastatin, we're prescribing all the other statins. I don't know if anyone actually is trying pitavastatin first, so the answer is gonna be.
Right, but hey, yeah, sorry, but I mean, I think to the original question, I mean, my patients, even the reluctant ones, once I tell them that I believe in it strongly and I think it will really help them, my patients' uptake has been excellent. And yeah, it's not pitavastatin, but it's a statin. So,
Right.
And then of course I have the rare one who said, still says no, and I respect their choice. That's their right. So,
Yeah.
Anyway.
Good to hear. Thank you.
That's wonderful to hear. I think, I guess my population skew younger and the conversations are often more difficult about, this is something. Yeah, we're not saying you're gonna take this briefly. This is this is gonna be a medication you take,
But
forever.
Jill, I'm not even offering it to patients under the age of 40. I chose the model of like a type 1 diabetic, and I spoke to endocrinology, and they don't start type 1 diabetics on a statin before the age of 40. So I don't know if that'll help you.
This is sorry, we went off topic on this conversation. Sorry.
Good conversation and good for me to hear, because I'm sure when I present this, someone else will have this question. So
Yeah.
at least I know how to answer it next time.
I don't know if anyone else has any comments. Actually, this is a great way to wrap up because we have a few minutes so we can have our dance party that I promised people we'd have here for the last few minutes. So I didn't announce it. If it works, it works. Just we'll put some music on at the end, but I did also want to make sure folks were aware. Sanjay, I'm sorry, I was gonna try to pull up the email. I know you're presenting next week. Can you do a little bragging about yourself? Or saying what you're gonna be talking about at Grand Rounds?
It'll be a surprise. Can I do that?
It's not a surprise though, right? It has a
It's
title.
not. It's not.
No, I'm sorry.
Oh, for Grand Rounds,
Grand
sorry.
Rounds, yes. I can do both. Yes, what are you presenting on next week?
It's just going to be the stuff that's we've been doing for a while, just molecular epi, but just the newest stuff that we've been working on. So
I think the title is Molecular Epidemiology for HIV Prevention. Correct?
that sounds right. Yeah.
So that's for regular medicine Grand Rounds. If people are interested, that's Wednesday at noon, and then next Friday, Joel Wertheim is presenting. Do you have
Oh, yeah,
the title?
it is. The rise and fall of the 2022 Mpox outbreak in New York City and its relationship to HIV transmission networks.
Good plugs, very exciting. Anyway, Sarah, thank you so much. I think we really, it's great to go back to your roots again with you and hear the journey and hope that it continues to go
It
well.
is exciting to see where I started and where it ended, right? It doesn't happen often. It might not never happen to me again to get to the clinical trial.
It will. All right, thanks, everybody. We'll next week.